Okay, good evening everybody, and welcome to tonight's Research Tuesday, which is called Sugar and Splice, as you can see. My name's Bob Hill, I'm the Executive Dean of the Faculty of Sciences here. I'd first like to acknowledge the Kaurna people, the original custodians of the Adelaide Plains and the land on which the University of Adelaide's campuses at North Terrace, Waite and Roseworthy are built. Tonight we're going to take a look at glycoscience, an innovative research area that examines the structure and function of carbohydrates, or more simply sugars. In August this year, the University of Adelaide launched a new centre, Adelaide Glycomics. Based at our Waite campus, this centre will be the largest facility of its type for the analysis of complex carbohydrates in the Southern Hemisphere. Our speaker tonight, Professor Vincent Bellone, is director of this new centre, and tonight he's going to introduce us to the field of glycoscience and share some examples of the applications and benefits of this research. Vincent joined the university in 2015 as Director of the ARC Centre of Excellence in Plant Cell Walls, a role that he still holds. Prior to his recruitment in Adelaide, Vincent was Director of the Centre for Biomimetic Fibre Engineering and the Advanced Carbohydrate Materials Consortium of the Royal Institute of Technology, a leading technical university in Stockholm, Sweden. Vincent is founder of the spin-out company Carbozide Proprietary Limited in Adelaide. The company will exploit his recent invention on UV protective materials based on carbohydrates and natural molecular sunscreens from algae. Following Vincent's presentation tonight, we'll invite questions from the audience, and I do encourage you to get involved. But first, please join me in welcoming Professor Vincent Bellone.
Well, thank you, Bob, for your very kind introduction, and welcome everyone to this Research Tuesday's lecture tonight. It is my great pleasure to be here and to introduce some of our research activities to the general public. So my lecture, as Bob said, is about carbohydrates and what we can do with carbohydrates. So what I will do is that I will start by giving a little bit of background about this type of molecules, how important they are, and then hopefully this background will be very helpful to understand the type of applications that I will present in the second part of the lecture, explaining how we can exploit carbohydrates and also enzymes that synthesize carbohydrates in different applications. So carbohydrates occur in nature and in living organisms in different forms. The simplest form is what we call sugars, the simple sugars, or monosaccharides, like glucose, for instance. And glucose is one of the most abundant monosaccharides or sugar in living matters. These simple sugars can also attach to each other to form more complex molecules, chains of sugars, that we call oligosaccharide if they are rather low number of sugars, or polysaccharides if we get longer chains. And these polysaccharides are also called complex carbohydrates or glycans. All these terms are synonyms. They can be applied and used in many different areas, as quoted here. And I will try in this presentation to give you some examples, concrete examples of applications in some of these areas. A very important discovery that was made quite a while ago was the discovery of molecules called nucleotide sugars. And these molecules were studied by Leloir, and he got the Nobel Prize in 1970 for this discovery. He was at that time studying a disease called galactosemia, and he could show that these molecules are very important in this disease. And later, it was shown that these nucleotide sugars occur, in fact, in all living cells, in all organisms. So it was a very important discovery, and they are the precursors of polysaccharides or complex carbohydrates. So please remember Leloir and nucleotide sugars. I will come back to this in a little while. The process that leads to the formation of carbohydrates, complex carbohydrates, is called the glycosylation. And glycosylation occurs in every single types of cells in nature, and it is a vital process. Without glycosylation, there would be no life. Basically, all types of cells are able to produce and form some types of glycosylation reactions. Most cells, with the exception of animal cells and small bacteria called mycoplasma, are in fact surrounded by a cell wall, which is the most outer layer of the cell and which is rich in carbohydrates, complex carbohydrates. Animal cells do not have really a cell wall, but they have something that is not quite as organized as a cell wall instead, and that we call the glycocalyx. So for instance, here you have intestine cells that show here at the top their glycocalyx. This is again a layer of complex carbohydrates or glycans. It is not as organized as the type of cell wall that you would find in bacteria, algae, or plants, but it is still something that is somehow comparable. There is virtually an unlimited number of possible structures of carbohydrates that could occur in nature. But nature is a fantastic engineer, and it has been able to select only, amongst all the possible theoretical structures, only the structures that provide excellent function and properties in different forms of life. And I will take some examples of this in this lecture. Every day in our life, we are confronted, or exposed, I should say, to different forms of carbohydrates from different sources. There is almost not a single moment in our life where we are not exposed to carbohydrates in one way or another. Cellulose is a very important polysaccharide that is a constituent of wood, and we use wood, of course, on many different applications, paper, for instance. The cotton fiber is also made of cellulose, carbohydrate. We eat a lot of carbohydrates in our food, and we derive a number of products that we enjoy, like wine, for instance, where the carbohydrate composition plays a role in the quality of the wine. Carbohydrates can also be used for concrete applications, like bioplastics. They're used a lot in the cosmetics industry to make hydrogels, but also creams and all kinds of cosmetic products. They can be used to make packaging material, and they can also be degraded and converted into the simple sugars I was talking about at the beginning, like glucose, that can then be fermented into ethanol and used as a liquid transportation fuel. They're also very important in the medical field. Here you have a schematic representation of a cell, of a human cell, and you can see that at the surface of the cell, many types of interactions involve carbohydrates. For instance, some pathogens, like some pathogenic bacteria, interact through carbohydrate interactions with proteins at the surface of the cell. Some diseases, like cancer, can also be initiated through interactions between proteins that are at the surface of the cell and carbohydrate molecules. And if we can find a way to interfere with these interactions, we may have a way to control and prevent the disease. So carbohydrates are also very important in the medical field, and there's a lot of research being done in that area. If you look at the composition of a typical cell, this is in fact a bacterial cell, but the same type of composition is taking place for animal cells and virtually all kinds of cells, with some differences in the relative proportion in the different components, organic components presented here. But the major constituent, in fact, of life is water. And in addition to water, there are a number of organic molecules, 4 main classes. The nucleic acids, which codes for the proteins that are involved and catalyze the chemical reaction that take place in our cells, and the lipids that form the biological membranes, and the carbohydrates. Carbohydrates are used as the main source of energy through the breakdown of glucose into energy. They're also used for structural roles, and they also have many other functions, and I will try to describe some of them or illustrate some of them in the lecture. So a little bit of background about carbohydrate structure. This is needed to understand better the applications I will discuss in the second part of the lecture. So if I would take a powder of glucose and dissolve it in water, in fact, the glucose molecule would take different forms. It can be linear as represented here, but it can also form cyclic forms in the form of these pentagonal structures, which we call furanols, or these hexagonal structures, which we call pyranose ring. And these different forms of glucose coexist in solution in water. But as you can see, they are equilibrium here with arrows that tend to point towards this type of structure. And this means that if you dissolve glucose in water, these two forms here, the pyranose rings, are the most abundant. But all the other forms exist, and they can be converted into one another through these different equilibria. So once you have dissolved glucose in water, and the molecule is cyclized like this, it can take different shapes in space, different conformation. And these different shapes go from one to another, and the most stable is the chair conformation. This is a little bit like our body can take different postures, if you like, an almost unlimited number of postures. So the sugar molecules can do the same. But the sugar molecules can be free, like in this example, but they can also be polymerized and attached to each other to form more complex structures. And these more complex structures are formed by a class of enzymes that we call glycosyltransferases. And they are in fact called Leloir glycosyltransferases because the substrates that they use are the nucleotide sugars that were discovered by Leloir, remember, at the beginning of the lecture. So what do these enzymes do? In fact, the reaction they catalyze is simply, as the name implies, the transfer of a glycosyl, a sugar, from the donor molecule, the nucleotide sugar, to an acceptor. And I will illustrate this with one concrete example represented here. If we consider cellulose, it's a polymer of glucose that are linked through these chemical bonds. And cellulose, this chain, can be elongated by an enzyme called cellulose synthase, which is a Leloir glycosyltransferase, and that uses UDP glucose as a substrate, the nucleotide sugar. And what the enzyme does is that it transfers this glucose residue on the chain to elongate the chain by 1 unit at a time. And it can do that in a repetitive way to make a very long chain of cellulose, which can be several thousands of glucose units linked together. UDP glucose is a very important nucleotide sugar, but it can be converted into different other nucleotide sugars. And in this gray box here, you have the different types of nucleotide sugars that occur in plants, in plant cells, and the enzymes that are numbered that catalyze these different reactions that allow the interconversion of these nucleotide sugars into one another. These molecules are extremely important because they are the precursors of the final polysaccharide that form the carbohydrates, the carbohydrate composites in the cell wall of the plants. The reactions, again, are catalyzed by Leloir glycosyltransferases. We just saw how cellulose is synthesized, but the other polymers, like the pectins that are found in plant cell walls, and these polymers here are synthesized using the same principle as I explained with the cellulose synthase example. These enzymes can form very complex structures. Here is the example of pectins. Compared to cellulose, it's a lot more complex because it consists of many more different sugars that are color-coded here, also with different shapes. So there is always a main chain, which consists here of these red and blue sugars, if I can say it like this. But this main chain in pectins can contain branches with additional chains that attach to the main chain. And this creates a very big molecule, which is quite heterogeneous and complex in structure, but that has very interesting properties. It can form gels. And we exploit these molecules in many food products, for instance, because it can be used as an additive to control the gelling properties, for instance, of jam. Another last example of the complexity of carbohydrates is what we find in cartilage, for instance, and which we call proteoglycan. So in these big structures, there's one part which is a protein part and the carbohydrate part, which is the glycan. These structures can be very complex, and the result of the polymerization of this type of units here in very long chains, a little bit like in the case of cellulose. And these chains that are represented here by these blue and red lines, so each one of them is one polymer of this type of units. As you can see, many can assemble together to form this type of very big complex structures, which all together form something even bigger. And this is a main constituent of our cartilage. And because these molecules contain a lot of these hydroxyl groups, they have a tendency to interact a lot with water. And this type of complex structure retain a lot of water. But they also have exceptional mechanical properties which are actually conferred to the tissue that is rich in this type of structures. Cartilage has some elasticity, but it's also mechanically strong. And these molecules, or super large complexes, contribute a lot to these properties. In plants, cellulose play that role together with other polymers. It's only one of the structural components that provides strength and mechanical support. Again, here one chain of cellulose, but in fact, in the plant cell wall, each of these chains interact together through multiple weak energy bonds. So if you would take the type of bonds that link the different chains together, they're very weak if you take them individually. But because the chain is very long, several thousand, and because there are so many of these weak bonds, the final structure is really strong. And in fact, if you would take one of these fibrils represented here, and if you would make a string of steel of the same diameter of this fibril, in fact, you would find that cellulose is stronger than steel. It would take less energy to break the string of steel if you apply a force along the axis of the string of steel than you would need to break cellulose. And this means that cellulose has remarkable mechanical properties that can be exploited in a whole range of products. And there's no way I can give an exhaustive list or an exhaustive series of applications here. I will just take a few examples to illustrate what we can do with cellulose. The first one is, again, related to the structure. If you would, for instance, take a single cotton fiber from your cotton shirt and be able to look into it in more details, it would look like this. It's, in fact, an aggregation of multiple fibrils of cellulose that each consist of multiple chains of cellulose. So this is highly aggregated, and it's a strong material that we use to make textiles. But we can do other things with this material if we are able to disintegrate it into individual fibrils, into each one of these components here that are represented in white. And to do that, we can use mechanical shearing, or we can combine the use of enzymes with mechanical shearing. And if you do that, you can observe, under different types of microscopes, these fibrillated structures. If you mix them up, if you homogenize them in the presence of water, you can form hydrogels. This is completely green material. It comes directly from plant cell walls. You can also press dry this material and convert it into a nanopaper. And as you can see, you get a semi-transparent nanopaper that is bendable and that is super strong. And in fact, the reason why it has these properties, it's because of the way the fibrils are organized in the nanopaper compared to a normal type of paper that we use for writing. As if you look on this microscopy picture, this transparent material has a much better distribution of the microfibrils of cellulose. There is less roughness compared to the normal paper, and the porosity is much smaller. And all these properties, these attributes, altogether provide transparency and mechanical strength. This is really strong, and there's potential applications to use this as a coating for all kinds of display, for instance, again, with a completely recyclable material, cellulose. Another application of exactly the same material is to make foams. So instead of converting the microfibrillated cellulose into a nanopaper, as I just showed, you can simply lyophilize the material, freeze dry it. And if you do that, you obtain a three-dimensional foam that is super light. This material is as light as polystyrene that is used for packaging. But as you can see, it's super strong. It can take up to 1,000 times its own weight without collapsing at all. And most of it is actually air. There's in fact very little cellulose in that material. And because it's porous, it can be used to carry different kinds of compounds. So it can be used for pharmaceutical applications as the basis for a delivery system. And it has excellent insulating and shock absorbing capacity. So this can be used potentially to make sophisticated materials that have all these different properties. Another way to exploit cellulose from plant cell walls is to break it down into smaller elements. This is again the type of fibrils that you can isolate from a plant cell wall. But if you treat it with a strong acid, you can break it down into crystals that we call cellulose whiskers, because they're as stiff as whiskers from a cat. It's just an image. And you can see that these elements are crystalline, and they have a size which is rather homogeneous, 200 to 300 nanometers. If you make a suspension of this material in water, if the suspension is a diluted suspension, the whiskers will be randomly and homogeneously distributed in the liquid. And then you get something that is a clear suspension. If you increase the concentration, you will start forming a crystalline phase, a liquid crystalline phase. And this crystalline phase has interesting properties, which we call chiral nematic liquid crystalline phase. I will explain what that means in a second. So if we look at this liquid phase, and if we move from the bottom of the tube to the top, this is what happens with the orientation and organization of the whiskers. In a given plane, the whiskers all have roughly the same orientation. But as you move upwards in the tube, you can see that the orientation of the whiskers change from one plane to another progressively, and basically you get a kind of rotation of the whiskers as you move upwards. This material can be dried. If we evaporate or remove the water, the orientation of the whiskers will be preserved. And this will form regular geometric structures that interact with light in a specific way so that it generates colors. And this is a way, for instance, to produce iridescent films after drying. So the films will change color depending on the incidence of light on the object and how the light interferes and interacts with the cellulose whiskers that are packed in different layers in different orientations. If you add a little bit of salt before you do the drying, you can really make iridescent paper, or you can control the color of the paper depending on the type of salt that you've added. And this is a mimetic system. It's just reproducing what nature is doing very well, for instance, in some insects. So applications of this, well, you can think of iridescent paints, for instance, and many, many other applications. This is just one example. So you could use that to have a paint for a car, which would change color depending on the incidence of light on your car. Potentially fancy materials. Similar principle, a little bit different, inspired by nature. You know that many animals, in fact, are able to change color when they feel, for instance, threatened by a predator, or simply when they're stressed. And there are two ways to produce color. One way is to use pigments, and that's simply a chemical that will absorb selectively some radiations from the light and reflect other radiations that give the color. That's one way. The second way is to produce what we call structural colors. And this is what these animals are using. The structural colors result again from the occurrence of organized structures. They can be in 2 dimensions, they can be in 3 dimensions at the surface of the animals. And these structures are able to interact with water. And when they interact with water, if water goes in, it can change the space between these geometric structures. And when this happens, the material does not interact anymore the same way with the light, and this means that the material will change color because it interacts differently in different ways with the light. So what can we do with that? Why am I talking about this? In fact, you can reproduce this by using cellulose together with polymers. So here, cellulose has been combined with a synthetic polymer, and I'm just illustrating here exactly what I explained in a materials context. So what you see here is a regular orientation of cellulose together with its polymer that has been used to make a composite, and you can see that the spacing between the structures changes as you move from that direction to that direction. And you can see the impact this has on the colors. The principle is illustrated here. So what was done is that the type of cellulose whiskers, or cellulose nanocrystals, were mixed up with a polymer, synthetic polymer represented here in orange. And this polymer likes to interact with water, and it's miscible with cellulose. So if you mix it with cellulose, and then you add water, it will take up the water, and the space between each nanocrystal will increase because it will swell in water. And the pitch of the overall helix will also increase. And by changing this geometry, but still preserving a very organized orientation of these whiskers, we can change the color of the material. And you can see here what happens with 100% humidity. This is basically in water, compared to only 30% relative humidity. There's a change in the structure, and this is accompanied by a change in color. I have a little movie to show you what happens in real life. So here, the composite was made and converted into a film. On a black background, the color is green, and what has been done here is that water is being added. In fact, the color change that you will see would take around 90 minutes if you would just increase the relative humidity in the air. To go faster, we have added water to the sample. And you can see after the water came in that it's progressively changing color, going towards the brown, the yellow, and ultimately it will get completely transparent. And you have some reddish as well. Progressively, it will get completely transparent. And because it is transparent at the end, and on a black background, you don't see the material anymore. So what can we do, in fact, with this? We can make materials that change color depending on the relative humidity in the air. Again, we don't need to cover the material or dip it in water. Just the relative humidity in the air, the change in relative humidity, is enough to change the color. And this is illustrated here. If we start with 30% relative humidity and we increase, you can see we go towards brown, orange, and then completely transparent. But what is interesting is that this process is reversible. If we remove the water and we decrease the relative humidity, we can go back to the green. And you can see here that we can do that many times. There's no change in properties, no matter how many times you add water or remove water, or increase or reduce relative humidity. The other beauty of this system is that you don't necessarily need to cover the whole spectrum of colors. By carefully selecting the polymer and the amount of polymer, it is possible to work, for instance, only in the red area, or only in the blue area, or only in the green area, and just have different nuances of green, for instance. And this is something that can be used potentially also to make fancy materials that will just change color depending on the relative humidity. So you can imagine a paint, again, for your house that will change color depending on the weather. Or you can imagine a shampoo that will change. It's probably not the cup of tea of everyone, but you could have your hair changing color depending on the relative humidity in the environment. All right, this was about cellulose. Another example that I want to discuss is chitin. Chitin is another abundant carbohydrate found in nature, and it can be used for many different applications, and I will just focus in the first instance on crop protection. So what is chitin? It resembles cellulose. The only difference is that one of the atoms, the carbon atoms of the ring here, has this N-acetyl group. That's the difference between chitin and cellulose. The rest is similar. Chitin can be deacetylated. So these groups here can be removed to produce chitosan. So chitosan is a derivative of chitin. It's a non-acetylated form of chitin. And this is quite important because by doing so, you release these free amino groups that are extremely reactive chemically. And try to remember this. I will come back to that in a minute for some specific applications. This means that they can be readily modified by other things. So chitin normally occurs in the exoskeleton of insects and crustaceans, and it's a very abundant polymer. There are different types of applications, surgical threads and wood care products. But what I want to discuss here in the first instance is the importance of chitin in fungal pathogens. It's a component of the cell wall of microscopic fungi. And this is extremely important because if you look at the past decades, we have an increasing number of outbreaks of fungal emerging infectious diseases, both in animals, in red, and most importantly, in plants. And this is everywhere on every single continent around the world. And about 75% of these emerging infectious diseases are due to fungi. And if you look here, you have 5 important crops with 5 different pathogens. And in 2011, an estimation is that if we had been able to cure or to prevent the diseases caused by these pathogens on these crops, we would have been able to feed around 700 million more people on the globe, around 8.5% of the global population at that time. So these diseases are very important because they challenge our food sustainability. So what is very important is to be able, of course, to tackle these diseases. And we're interested in my research group essentially on this type of pathogen, which is not a true fungus like the other ones. This is called an oomycete, and it's responsible for the potato late blight, as illustrated here. This microorganism, this pathogen, can basically kill a whole field of potato within a few days, completely wipe out the whole field. A similar organism, which is also an oomycete, can infect salmon. And this pathogen can lead to up to 50% loss in the aquaculture industry of salmon. So it's a very important pathogen, economically speaking. And until recently, it was controlled, the disease was controlled by using malachite green. The problem with malachite green is that it accumulates in the tissues of the fish and it's converted into other compounds that are carcinogenic. So malachite green has been completely banned worldwide for use in aquaculture. So a consequence of this, of course, is that now we don't have any ways to control the disease. So what can we do to try to control the disease in these pathogens? Well, the cell wall and the carbohydrates are an excellent target, and I will try to illustrate this. This is the cell wall of one of these oomycete pathogens, and you can see the cell here. The shape of the cell is preserved. The cell wall consists essentially of these microfibrils. This is cellulose, and this is what distinguishes the oomycetes from true fungi. True fungi do not contain cellulose, but oomycetes do have cellulose in their cell walls, a little bit like a plant. But some species, like this fish pathogen, also have these small granules in their cell walls, which corresponds to chitin. There is very little, 1%, 2% at the most, very, very little. But this chitin, as you will see in a couple of minutes, plays a very important role in the stability of the cells of the pathogen. So in my group, we have identified and isolated the enzyme that is responsible for the biosynthesis of chitin. It is a chitin synthase. It is also a Leloir glycosyltransferase. Remember Leloir and the nucleotide sugars? So this enzyme uses a nucleotide sugar. And here we have been able to reconstitute the enzyme using recombinant DNA technology in an active form in vitro. And if I add to this enzyme the nucleotide sugars, it will form these small crystallites of chitin. In other words, we can reproduce in a test tube what the enzyme is doing in the cell of the pathogen. And this is an excellent tool then to test inhibitors of the enzyme. And this is the natural substrate of the enzyme, and this is an inhibitor that resembles a little bit the nucleotide sugar represented here. The molecule is different though. There is no sugar here. The chain is different compared to the natural substrate. This molecule, called nicomycin Z, sits very well in the catalytic center of the enzyme. In other words, it can replace the natural substrate, the UDP and acetylglucosamine. The consequence is that if the enzyme is mixed with this nicomycin Z, it's catalytically inactive. It can't work anymore. And if you block this enzyme, the chitin synthase, you can see that the pathogen at a certain concentration stops growing. So this is a way to kill the pathogen, and this is a way we can potentially control diseases caused by these pathogens. And this is what this video illustrates. So look at these cells here. These are the pathogen cells, and then this one. What we have done here was simply to add nicomycin Z to the preparation. And see what happens. The cells just burst here at the tip. I will play this again when it's finished, and they basically die. So what's happening when we do that is that we block the chitin synthase, the enzyme that forms chitin, and even though there's only 1 or 2% of chitin in the cell wall, the cell gets fragilized, and then the cell wall cannot sustain anymore the strong turgor pressure that comes from inside the cell. And therefore, the wall is fragilized. It cannot take this turgor pressure, and it basically just bursts. And this leads to cell death. These cells here are completely dead. So this is a way to kill the pathogen and to control the diseases. Another way to do that is to use a different approach. And I will explain what we have done here in a few minutes. This is a magnified leaf of a model plant species that we use in the laboratory. It's called Arabidopsis. And the leaves have at their surface this type of single cells that we call trichomes. And what we have done in this plant is that we have introduced the gene that is responsible for the formation of chitin. We have added to the plant the chitin synthase gene from the oomycete. And we have tried, by doing that, to make the plant produce chitin. Chitin is a polymer that does not occur at all in plants. But here we have tried to engineer the plants so that they would produce a little bit of chitin. And we were successful in doing so. These red dots here show the presence of chitin. Chitin is detected by 2 types of proteins which strongly bind to the chitin, abbreviated here by WGA for wheat germ agglutinin, and this is a chitin binding domain. And to visualize the binding, we use fluorescent molecules that give a red color. As you can see, this is the modified plant, and this is the non-modified plant. There is no red dots here, which means that indeed there is no chitin in these plants, but the modified plants do produce chitin. If not, you look at the surface of the leaf. This is the surface of the leaf of the non-modified plant. It's perfectly smooth. But the plants in which we have introduced our gene start producing these plaques, which at a higher magnification consist of these granular structures. This is in the modified plants. And these structures look exactly like what you have in a mollusk shell or a crab shell. It's chitin. So in other words, we have engineered plants that produce chitin, that produce a polymer that is normally produced by crustaceans or animals. And why did we do that? What is the consequence of this? Remember that pathogenic fungi and also some oomycetes have chitin in their cell wall. So now the plant is itself producing chitin. And the consequence of that is that the plant will react as if it was infected by the pathogen. And this will trigger the natural defense mechanisms of the plant as if the pathogen was present on the plant. And see what happens when we do indeed challenge the plant with the pathogen. This is the modified plant. It's perfectly healthy. It is able to resist against the infection. Whereas the non-modified plant, you can see the tissues are getting brownish when it's infected by this pathogen. It's basically dying. So this is a way to protect crops against pathogens. You can, of course, translate this in commercial crops. This was done in this Arabidopsis plant that we use in the lab. But you can do that in, for instance, cotton. And there are 2 advantages here. Not only your cotton will be more resistant to pathogens, but at the same time, it will contain a little bit of chitin that we can convert in chitosan. Remember, these very reactive amino groups. So what can we do with that? Well, now we have here a molecular tool that we can modify, for instance, with dyes and attach through these amino groups covalently, in a non-reversible manner, the dyes to the chitosan. And then if you make a textile of this, you end up with a textile that will never lose its color, no matter how many times you wash it. It's very important for the textile industry. But instead of attaching a dye, you could also attach an antibiotic or an antifungal compound. And then when you make a textile out of it, you get a textile that will keep fresh longer. You could attach a molecule that repels water, and then you get a self-cleaning textile. If you walk in the rain, it will just clean itself and let the water go. All right, another last application of chitin and chitosan that was developed in my group as well quite recently. We can use chitosan to make UV protective materials, and I will show you how. First of all, I'm sure you're all aware that it's not very good to overexpose your skin to UV radiations. There are some benefits if the dose is controlled and very carefully controlled, but the disadvantages are a lot worse than the benefits. So basically, it is very important to avoid overexposure to UV because this can induce skin cancer, which is a very big problem, of course, with the levels of UV radiations we have in Australia. There are very good filters against UV radiations already that are used in commercial products. The problem with these compounds is that they're not that friendly, and they're also usually combined with nanoparticles. The other thing is that sometimes you need to combine several to block both UV radiations, UVA radiations and UVB radiations. And there's more and more evidence that nanoparticles can get across the skin. And if some of these compounds get across your skin, they can provoke hormonal disorders, for instance. So if you keep applying a lot of these commercial creams with a lot of these filters, in the long term, it can have some effects on health. So what we thought we should do is to use something that is more friendly. And to do that, again, another kind of biomimetic approach was to search for natural compounds that are able to absorb UV radiations. And where do you go and find these compounds? You found them in environments that are naturally exposed to high UV doses. And some microalgae produce these compounds called mycosporins or mycosporin-like amino acids. And the fish that eat these algae end up with mycosporins in the lenses of their eyes and in their tissues. And this helps them to protect themselves against the UV radiation. So we have exploited this property to make interesting materials. There are quite a number of different molecules that occur in nature. And look at these numbers here. This is the most important on this slide. These numbers tell you how much these molecules are able to absorb UV radiation. The higher the number, the more powerful or the more able the molecule is to absorb UV radiations. And to give you an example, this is a value that is equivalent to twice of what you need to claim an efficient commercial product for UV protection. So they're twice as good as some of the products that are on the market. They also absorb both UVB and UVA. Therefore, you don't necessarily need to make complex mixtures of these molecules to get something efficient. So what we have done here was rather simple. Again, the chitosan, for instance, from crab shells, and remember again this very reactive amino group. What we have done was very simple. We used this reagent called a carbodiamide to make a covalent link to attach firmly the mycosporins, the UV-absorbing molecules, through their carboxylic function to the amino group. This is the result of this reaction. We form what is called an amide bond, which means that now the mycosporin is firmly attached to the chitosan. So we have made a solid physical material out of this. And this is the structure of the final material with the chitosan here and the UV-absorbing molecules. We have used 3 different types in this example. What can you do with that? You can convert the material into a film, and the film will be able to absorb both UVA and UVB, but at the same time, in the visible part of the spectrum, you can see that it is transparent to light. So we can make films. I have samples here. If someone is interested after the lecture, I could show you some concrete samples. Come to me at the end, and I will show you. So we have a film that blocks both UVA and UVB, and that is completely transparent. And we don't need necessarily to make a film. We can use this as an emulsion, as a component of a cream, as a suspension in different ways. What we have done here was to modify only 3% of the amino groups of chitosan, which means that we can put even more microsporins and get a complete blockage of UV light. It's a very efficient screen. But it has other interesting properties. So if we subject the film to very, very high doses of UV light, equivalent to what you would get outdoor, for instance, in Queensland, the material would be stable. If you would use it as a coating for a table and leave your table outdoors for 10 years, the material would be stable during the entire period. Only chitosan, if you just use chitosan represented in blue here, you can see some degradation over time. But the material that contains the microsporins is very stable. 10 years in Brisbane, and it's still stable after that. So we could have used even higher doses to see how far we can go. So these has potential applications for coating of furnitures, for instance, and all kinds of materials, outdoor textiles for your boats that you use in the summer, etc., etc. There are many, many potential applications. It's also resistant to heat. So if we cook the sample, in other words, there will be no impact on its capacity to absorb UVA and UVB. This is the chitosan control, but these are the modified materials with the microsporin. It is thermoresistant. So it's very good. If you have it in the sun, it will resist to the heat of the sun. Importantly, also, it is biocompatible, which means that you can grow human cells on this material. Here, we didn't use human cells in this experiment. We used cells from a mouse, but you can see that the cells can grow on the material. This is what we call a live dead type of assay, where basically if the cells are metabolically active, they are able to convert a chemical that we add to the cells into a green fluorophore. All the cells that are green are alive. If the cells are dead, they're red. You can see there's very, very, very few dead cells. This is an excellent material that is compatible with the growth of animal and human cells. And the cells stick very well to the film. You can see here that they attach to the film very well. And this is very important because it means that you can use this material. It's compatible for use in humans for all kinds of products that will protect against UV, but we can also convert this into multifunctional products by combining different properties, not only UV protection, but other properties as well, like antibacterial properties, for instance. So I'm almost done with the examples. I hope that with these examples I have shown you the importance and the relevance of carbohydrates in many applications in our daily life. I would like to finish with just a couple of slides to give you some perspectives. So the type of work I've presented can be used for many different things. And in our society, we're actually having and producing more and more waste from agriculture. And if you just look at the South Australian state, you can see here this number says that we are basically producing around 27 million tonnes per annum of total waste from agriculture. 27 million tonnes per annum. Out of this, around 20%, 5.5 million tonnes, can be recovered. We say they are accessible. This is something you can harvest. But look at what's happening here. Most, in fact, of the waste comes, or a lot comes from animal waste, but this cannot be easily recovered. It's left behind, we can't recover it. But the crop residues correspond to 95% of the accessible waste. And these crop residues are rich in carbohydrates. So what we want to do in the next step is to use some of the same principles as I have explained throughout the lecture to convert this agricultural waste into valuable products. At the moment, this is what we do with this waste. We burn it to produce energy. We use it for compost, and we use it for feed for animals. These products have very low value, and there are better ways to produce energy than burning this material, which is very valuable. I will just take one example relevant to the South Australian state, but there would be many others. Here I'm just going to talk about potato, but we could talk and use the same concept for waste from wheat, barley, all kinds of crops that we grow. So the South Australian state is the largest producer of fresh potato in Australia. 80% are produced in this state. And we still generate quite a lot of waste because you won't buy these potatoes if they go on the supermarket. You won't like them. They're ugly. They have wrong shapes. They don't have the right size. They're ugly. You don't want to buy them, right? So therefore, they're a waste. And just in the state, we produce around 32 million worth of waste every year. And the number is around 80% of what's happening in the whole of the country. Paradoxically enough, Australia imports potato starch. So we have all that waste, but we don't do anything with it. We just import potato starch. And the main reason for that is because there is no potato starch industry in Australia. Therefore, we can't use this waste for anything useful. And what could we use this for? Well, the potato is very rich in starch. Starch is a way to store energy for the plants, and animals have glycogen instead, which has very similar structure. And there are different forms of starch. This is, again, a carbohydrate. One form is amylose, which would consist of single chains like this that are free. Another form is amylopectin, where you can see lots of branches, a very complex structure. And the starch can take different forms depending on the type of plant you consider, and different shapes of the granules. The starch granules in potato, for instance, they're oval. In the rice, they have these angular shapes. What can we do with these different forms of starch? Well, we can do something more useful than just animal feed, for instance. We can convert it into different products. Amylose, one form of starch, is very good to make low glycemic index food. The so-called resistant starch is very healthy. What we call resistant starch is a form of starch that is not degraded in our small intestine, but that goes through all the way to the colon, where it is then degraded by the microflora, the bacteria that we have in the colon, and that can ferment this resistant starch into small fatty acids. Butyric acid is one of them, that has very healthy properties. It helps fighting against cancer. It helps fighting against the development of colon cancer and also against pathogens. So these different forms of starch can be used to make functional food and derive many food products instead of using them for the type of applications that have very low value that I mentioned earlier. And the type of starch that we can't use for food can be turned into bioplastics, for instance, and can be used for many different materials. So this is one area where we're going, trying to convert this increasing agricultural waste into valuable products. And why am I saying that this agricultural waste is increasing in terms of amounts? It's because the population is increasing. Therefore, we need more agriculture. And we have more agriculture. Of course, we need agriculture to feed ourselves, but we also generate more waste. And with this waste, we can do very valuable things. This is just an example. I could go on like that with many other crops and other things that we can do. But I will stop here, and before I completely finish, I would like to thank all my students and postdocs and researchers that have worked with me, and a lot of national and international collaborators. I can't list of them on this slide. There would be too many, but I would like to mention just a couple of names. Dr. Chitsu and Kuhn have contributed to the materials aspect that I've presented. And one of my talented PhD students, Elijah Shutek, together with Sarah and others, have done quite a lot of work on the oomycete. And with this, I thank you for your attention.